Hi, my name is Adam Cohen. I'm a hematologist-oncologist and director of myeloma immunotherapy at the University of Pennsylvania in Philadelphia. Today I'm going to be talking about a phase 1 trial of P-BCMA-101, which is a B-cell maturation antigen-directed CAR T-cell product for patients with relapsed and refractory multiple myeloma. This is a multi-center study, and the data are being presented by my co-investigator, Dr. Krina Patel from MD Anderson. So this is really a unique CAR T-cell product that's a little bit different from some of the other ones that have been presented over the last couple of years and at this meeting. The method of getting the CAR, BCMA-specific CAR, into the T-cells uses something called a transposon, which is a little different than the typical viral vectors that have been used in other trials. This allows for a potentially safer and less expensive process. It also has a larger capacity to insert multiple genes. And so in addition to the CAR recognizing BCMA, there is a safety switch that's involved that allows the CAR cells to be turned off if there's too much toxicity. And there's also a selection gene that allows the CAR product in the end to become very purified. In addition, this transposon-based process leads to a CAR T-cell product that has a higher proportion of central memory and also stem cell memory-like T-cells that are then infused into the patient. This potentially has the capacity then to have more long-lived CAR T-cells once the T-cells are infused, which may lead to better persistence. And so in this phase 1 trial, patients had a median of 5 prior lines of therapy. And all patients have T-cells collected and manufactured, and then they can get additional bridging therapy while the manufacturing process takes place. They then are given fludarabine and cyclophosphamide as conditioning, and then the CAR T-cells are infused as a single infusion. This is a dose escalation study, and we've now tested 5 different dose cohorts ranging from 50 million CAR T-cells all the way up to over 1.1 billion CAR T-cells. At the presentation here at ASH, there have been 23 patients so far who have been treated, and 21 are evaluable for safety. And what's really striking about this product has been the lack of cytokine release syndrome compared to other CAR T-cell products. Only 2 patients out of the 21 so far that are evaluable have had cytokine release syndrome, both of which were low grade, and only 1 patient has had any neurotoxicity. And therefore only 1 patient has received the anti-IL-6 drug tocilizumab out of all the patients. The other interesting difference in this CAR trial so far has been that the CAR T-cells seem to expand a bit more slowly. They peak around day 14 to 21 compared to day 7 to 14 in most prior studies, consistent with the fact that this may be a somewhat different T-cell type of product. In addition to the good safety profile, we are seeing activity in these heavily pretreated patients. So far, 12 out of 19 patients evaluable for a response have had at least a partial response or better, including some patients with stringent complete remissions that are MRD negative. And as we're now getting up into the higher dose levels, we're hoping that that response rate continues to go up. And so the trial is ongoing right now. Once the appropriate dose is identified, there's a plan to expand to a phase 2 to try to get a better sense of safety and efficacy in this heavily pretreated population. The study continues to accrue. It's currently open at our center at UPenn, also at MD Anderson, Sarah Cannon in Nashville, University of California, San Diego, the Colorado Blood Institute, Johns Hopkins University.